First, the signal proteins bind to the tyrosine kinase receptor. This causes the two receptors to come together to form a dimer. Then ATP of the dimer changes to ADP after a phosphate group is removed and placed on the receptor. Inactive relay proteins bind to the phosphates and become active. This causes the relay proteins to move away from the tyrosine kinase and change shape. This signal transduction pathway causes the ER to receive a message and send out a cellular response that releases calcium ions from the ER.
First, the signal protein binds to the G protein link receptor. This causes the attached inactive protein to become active. GDP of the G protein gains a phosphate and becomes GTP. The protein then moves away from the receptor and binds to an inactive enzyme, causing it to become active. Then a phosphate is removed from the protein and placed on the enzyme, changing GTP to GDP. This sets off the signal transduction pathway, which releases a phosphate to be sent to the ER. This activates a cellular response, which triggers the release of calcium ions.
The neuron has vesicles that contain dopamine, which are released by the cell. They bind to the dopamine receptors on the dendrite of the receiving cell. This sends a signal that releases dopamine into the cell. The neuron has vesicles that contain dopamine and meth. These molecules are released by the cell, and the dopamine binds to the receptors of the dendrite of the receiving cell. Due to the meth, this triggers the release of too many dopamine molecules. Meth also blocks the dopamine channel, which makes the cell unable to reuptake dopamine.
Symptoms of meth include irregular dopamine levels, addiction, hallucinations, problem with motor activity, changes in brain structure and neural connections, aggression, and dental problems. Treatments include behavioral therapy, medications such as AV411 or antidepressants, and stopping the use of meth, which can help dopamine levels become more regular after 6 to 12 months.